This was a fun presentation to put together because the instructions said, you can go back in time and pull together all the relevant work you've done on this topic. And we've been working on this for quite some time. And it was fun to kind of put those ideas back into perspective, especially given the new computational capabilities that have come online with AI and compute power. So let me jump right in. So the basic idea that we sought to address when we approached this problem was to be able to develop a generalizable framework that we could apply to any complex system. We started with microbial communities, and in particular on specific understudied microbes. And we started with a microbe called Halobacterium. And our objective was to decipher the gene regulatory networks of this microbe de novo, meaning there's very little information known for this microbe, and this would be true for a lot of organisms then and even now. So we wanted a data-driven approach to this discovery. So we generated a lot of data from which we developed these models that I'll talk more about. And these models are based in causation and mechanism. So they, in principle, were intended to drive some rational approaches to doing synthetic biology, discovering new biology, and so forth. And I'll give you some concrete examples of that. The second objective linked to this, I said it needs to be generalizable. What I meant was, while we're applying this to microbes, we wanted the approach to be generalizable to higher order complexity of multicellular systems, including host pathogen interactions, human disease. We worked on brain cancer and multiple myeloma, and we've also applied it to the context of ocean acidification and climate change, in particular to corals and to diatoms. And I won't say much about that, but I think that idea of being able to apply a capability that builds on some foundational evolutionary principles makes all of this possible. So the basic idea, regardless of what system you go with, our thinking was, this is the classic perturb, observe, I don't know why that's shifting, perturb, observe, and model, meaning you have a biological system for which, when we started with Halobacterium, and for a lot of the new systems we're applying to, the situation is the same. Very little biology. And we have the genome sequence, we know the genes, we know the proteins. We don't know how they operate, who they operate with, and in what context. So you would take a black box with just the genome information and subject it to ecologically relevant perturbations and observe responses of the system by doing multi-omic profiling, transcriptomic profiling being the major type of data that we had measured. And then you do a reverse engineering step where you try to reverse engineer the topology of the regulatory network that causally and mechanistically underpins the responses that were observed. And you do it in a way that I'll describe, but the idea here is the causation and mechanism that you incorporate into the building of the model allows you to then make predictions of responses to new experiments that were not used to train the model, and also gives you guidance in doing targeted perturbations so you can manipulate the behaviors of the system. So one of the key steps of this approach is to figure out modules. And this is very relevant to this group today. And the way we defined modules at the time, and it is still relevant, is the context in which subsets of genes get co-regulated by specific mechanisms. And de novo, you don't know what those genes are, so you would do this in a bootstrap reverse engineered approach where you would seed the clustering with some set of genes, and then you let the algorithm then explore the space of the rest of the data using some prior knowledge, such as possible gene-gene associations or interactions that you might have, even through computational approaches like phylogenetic profiling and so on, to help the algorithm identify which genes are co-regulated. So these are co-regulation patterns to the left of the vertical dashed line, and context in which then they are not as dynamically changing or co-regulated. And this is a zoom in into the co-regulated contexts. And we say co-regulated because this iterative approach is essentially constantly cross-validating to see if there's evidence of regulatory mechanisms. Here we're looking at regulatory motifs, cis-acting regulatory motifs in the promoter of the gene. But we've also looked at enrichment of known active binding sites when we've applied this to more complex systems. And you can think of generalizing this to also microRNA regulation, to post-translational or post epigenetic regulation, essentially modification of the genome and so forth. So this is a bicluster. This would constitute one module. And here you have information on which genes are in this module, what regulatory motif is likely mediating their regulation. It gives you the context in which they're being regulated, and some other information regarding their functional association. Now, we can discover in a single run for a simple microbe hundreds to thousands of modules. And the property of the modules is such that a gene can be part of more than one module, because we know genes can participate in multiple processes inside a cell. And for a given module, we then ask what are the factors that drive its co-regulation of the member genes. And you apply some differential equation type modeling, and you identify the factors, and you figure out a linear combination of both environmental and transcriptional factors that explain the observed change. And you use generalized linear modeling here. And you generate models that, in this case, for example, tell you that there's about 34 genes in this module, or bicluster 66, that are regulated by 2 transcription factors and 2 environmental factors. And one of the influences is conditional on light. And now we can just take the measured values of the input factors, and we can predict the relative changes of genes within the module. And you can look at the predicted profile is in blue, and the measured profile here is in red, and you see they're quite correlated. And in fact, you can also see that this holds true in new experiments, meaning these were held out and not used to train the model. And these were also not related to the specific environmental contexts that were used to generate the training data set. And this is shown at the level of an individual module, but this is also true at a system-wide scale, something we showed in 2007, where we show that you can make predictions of gene expression changes across nearly 80 to 90% of the genome of this organism. Subsequently, we've shown for Mycobacterium tuberculosis and other systems, you can go up to 93% or higher. And the performance of the model is indistinguishable over the training and the test data sets. Now, this is a hypothesis generator. And I'll get to some of those details in a bit in terms of how humans can use this model and how we can also use this in a more quantitative framework to predict phenotype. Okay. Now, what I showed you is the modeling done 20 years ago, but what we were aware of the shortcomings of the model was that there were several. One was that because this is a biclustering problem, as all of you might appreciate, it's an NP-complete problem. So every time we run the model, the composition of genes and environments in given biclusters are not identical from one run to the next. So the gene membership in modules is not statistically defined. I'll give you an example of that in a minute. And while we are identifying promoter architecture, some of the mechanistic details are lacking in terms of being able to specifically associate transcription factors to specific locations at nucleotide resolution. And also, if you have a very large data set, and the problem gets worse as your data set becomes even larger, is regulatory events that happen in some rare contexts. And I'll give you some examples later on, but I'll mention one. So, for example, when Mycobacterium tuberculosis infects the host, it is engulfed in macrophages and it lives inside phagosomes. Phagosomes are really difficult to profile. You get a few transcriptome profiles from that condition, but regulation in that context is really important. So how do you identify regulatory mechanisms that occur in such rare contexts? And we knew that the model, the way we had built it originally, was falling short of that. And it also did not predict in what context the regulatory mechanisms were active in a probabilistic way. So we can say that this module is functionally active in low pH and is being regulated by this particular regulator binding to this particular regulatory element. So that level of detail would help enormously in kind of increasing the likelihood of finding biologically meaningful regulatory patterns for experimental validation. And the final thing we knew was this is a regulatory model. It does not have direct tie-ins into phenotype. And so knowing the regulatory patterns would require an interpretation by a human to then design experiments to test what those regulatory events really meant. So we could change this through integration with metabolism, for example. So I'll cover some of these points as to how we address them. So as I mentioned, when you run the biclustering algorithm, originally it was called CMonkey for cluster monkey, and there was a CMonkey 2 version, and we have better versions now to discover these biclusters. So if you run this multiple times on the same data set, what you find is that you find a similar bicluster in each run, but it's never identical. Okay, and if you squint your eyes and you're able to read some of these gene names, there's a lot of overlap between these genes. And if you look at the motifs, you'll see like this motif, the number 2 in this fourth cluster, looks similar to motif number 1 in the third cluster, and in the first cluster, and in the third cluster is motif number 3. And they all look slightly different. Now, the differences are because of the slight variations in gene composition in each module. Now, this could be technical variation, or it could also have biological meaning. What I imply by that is, in some contexts, you may have a unique composition of a module, and in that context, the regulatory logic that is operating is slightly different. So you have subtle differences in the conservation of the cis regulatory motif for the genes in that particular module, and so on. So to figure out whether it's technical or it's biological, we need to have some probabilistic assessment. And for that reason, we developed this approach, which what we called at that time framework for inference of network ensembles. Basic idea here is you run biclustering and the network inference hundreds of times. But what you also do is you don't run them on exactly the same data sets. You take subsets of data, and you can subset the data in a meaningful way. So you can combine all the experiments related to redox and oxygen stress, high and low oxygen transition, et cetera, into one group. You can group together all the experiments related to low pH or low nutritional conditions or stress-related conditions as one subset. Or you can, and you can also have these data sets recombined in stochastic ways. You can just randomly subset the data because we don't know everything about how complex environments changed in the experiments that were conducted. The whole idea here is by subsetting these data, you increasing the power of detecting regulatory patterns that may happen over subsets of experiments and not all experiments. And I'll show you examples of where we really benefited from doing this. So the consequence of this ensemble run is hundreds to thousands of biclusters, and nowadays we're generating millions of biclusters from a very large compendium of data that has been subsetted in different ways. And this represents a space of associations amongst different components. So genes, it tells you which 2 genes are likely co-regulated, in what contexts, by what transcription factors and which environmental factors, through which cis-acting regulatory mechanism. Okay? And you can look at the frequency of co-occurrence patterns that are expected at random, and then you can calculate the probability of observing actual co-occurrence patterns to assign confidence values to these entities we call co-regulated modules. So this is the foundation on which we have a much better definition of a module, which we now call co-regulated module or conditionally co-regulated module. And it essentially is a module of genes that have evidence of being co-regulated in a defined set of environmental conditions by some defined factors, transcriptional and environmental factors, through specifically genomically located regulatory motifs, which we call gene regulatory elements. And I'll expound on that a little bit more. But when the context changes, the modularity also changes dynamically. So some genes, like these 2 genes here, break away, and these 3 genes now come in and merge with these prior genes to form a new module. And that happens over these contexts. And you can see there's an overlap of context, and both modules are operating indistinguishably. This is a very fundamentally important concept, especially when it applies to things like metabolism, where pathways are branched, and they can be broken in some contexts. They can reverse directions in some contexts, and they can essentially come together in other contexts. And I'll show you examples of nucleated metabolism where that might happen. So this type of plasticity that you observe at the phenotypic level is governed by parallel plasticity of the modularity, the dynamics of modularity at the regulatory mechanism level. And here's one example. And if you're a microbiologist, you'll appreciate that when genes are in an operon and of a related function, we often conclude that they constitute what is called a polycistronic transcript, meaning a single RNA traverses through this set of 6 genes to give you one transcript that has information for translating 6 proteins. Okay, and these are proteins, genes related to peptide transport, and you can see that in the bottom. This operon is in the reverse standard, and that's the reason I'm going from right to left here. Now, conventionally, you would look at something like this and just conclude that this is a single transcript. But when you look at actual data, there's a lot more complexity. So at the very top here, I'm showing you how we map gene regulatory elements through the approach that I'll describe in more detail later. But we've identified these 3 regulatory elements, GRE1, 17, and 10. They're located here in the middle of the operon. Okay, whereas in the very beginning of the operon, in the first gene, upstream to the first gene, you see a couple of, or one similar gene regulatory element, number 17, but there's a completely new one that also emerges here. And then you can look at the dynamics of how exactly this operon is transcribed. And this is essentially looking at signal going from early log phase, as you can see here in panel B, to stationary phase. So in the early phase of growth, you see signal throughout this whole operon. So it appears to be a single transcript. But when the cells go into mid to late log phase, there's a break that appears right here. So transcription terminates over here. And then as you go further up into the growth curve, you see that transcription comes back up for some of these downstream genes. So how is this accomplished at the mechanistic level? And you can see it's partly explained by these locations of these transcripts in the promoters that I mentioned earlier. But you can go down and you can look at the raw transcript structure and get one more piece of evidence that there is a segmentation here, which means the signal from raw abundance reads also suggests that there's at least 2 transcripts. But the regulatory network modeling has dissected this further and given us insights into 3 isoforms of transcripts from a single operon. So you have the red transcript that covers all genes, then the navy blue transcript that covers the downstream genes, and then the green transcript, which is covering just the first gene. And you can actually see where these genes fall in terms of regulatory patterns. And they have very different behaviors in terms of what kinds of corems they group into. So the full transcript here is a member of this corem 21645 that is primarily governed by the activity at the regulatory element number 17, which I showed you, these 2 elements here. So even though it's a single transcript here, what this analysis is suggesting is that there might be 2 regulatory events that are indistinguishable, but you're getting a similar regulatory pattern for all the 6 genes. But the second transcript, the blue transcript, falls into a different corem, and then the green transcript falls into yet another different corem. In each case, you can assign the mechanism that is responsible for splitting the operon, and it gives you what functions are associated with the other members of the genes in each of the corems. And you see functionally, this gene is getting involved. These genes are getting involved in slightly different processes depending on context, and they're doing so in different conditions. Right? So this module has got very rich description of composition, context, mechanism, and likely functional implications. Okay. And so how do we get to these gene regulatory elements? And I mentioned this earlier, that when we do biclustering, we look for cis-regulatory motifs, these CRMs or cis-regulatory motifs, that also are discovered in slight variations across the hundreds to thousands of ensemble runs that we have done. Now we can essentially find out which ones are similar to each other and do clustering of the motifs to identify these clouds of related cis-regulatory motifs, which we call gene regulatory elements. And this is how we get a dictionary of the regulatory mechanisms, cis-acting regulatory mechanisms across the entire genome. But the thing to remember here is we've done this by biasing the discovery process to the motif discovery process to occur upstream to the first gene of an operon or the first gene of a singleton, okay, but within 200 nucleotides or so. So that's because we are constrained by what we've learned through conventional approaches of where regulation occurs in prokaryotic systems, although we know that there's exceptions to this rule. And so for this purpose, we never really find new regulatory mechanisms inside genes or inside operons. And we can get around that problem by taking these regulatory motifs. So here I'm just giving you an example of multiple cis regulatory motifs that look similar, but there's slight variations. And when you group them together, you get a very strong signal for a gene regulatory element, which this one is a cluster of 700 motifs. And these are this is of 300 motifs. So what we can now do is, now that we've identified the regulatory motifs, which I should also mention that computationally it is easier to find motifs in a location where they're more likely to occur because they're small words and their co-occurrence frequency can be very close to the co-occurrence or the occurrence of random sequences of similar length. So when you bias the discovery to short regions that are in front of genes that are co-expressed, your likelihood of finding these motifs is very high. But then again, you bias in terms of where you found the motifs. Now, to find motifs elsewhere in the genome in non-canonical locations, we take these CRMs and we just use an algorithm called MAST, which is one of the algorithms in the meme suite, and we find out where in the genomes these cis-regulatory motifs align. Okay, at any given location, we might find that there's a large number of cis-regulatory motifs that are aligning at the same locations. And we also find from a clustering analysis that these cis-regulatory motifs are all members of GREX. Now what we can do is at that given location, because we're trying to differentiate technical from biological information, at each position, we can look at how many distinct CRMs that align to that position. We can backtrack and identify what were the contexts in which those CRMs were discovered. So we can then associate from the occurrence frequency of the cis-regulatory motifs, the likely activity patterns for the GRE in that location. I know that's a lot of words, but it'll become simpler when I show you this. So here's an example of, this is the carbonyl phosphate synthase gene. It's a key gene that sits in the center of amino acid metabolism and nucleotide metabolism, and there's multiple branch points. This gene is under control of many different regulatory elements. Each of these colored histograms refer to a binding site for a particular transcription factor. And I'm showing you two well-characterized ones, and the other ones match to other factors I'm not showing right now. So just through discovery approach, we have uncovered a combinatorial regulatory scheme of up to 8 to 10 transcription factors, including PRR, which is a well-known purine metabolism regulator, and RJAR, a well-known arginine metabolism regulator. What we can do is now look at the dynamic activity patterns of these regulatory motifs across contexts. Okay, so we're essentially cycling through different contexts where, as I mentioned earlier, the frequency pattern of alignment of CRMs is distinct. Okay, so PRR motif is active in some contexts, RJAR motif is more active in some other contexts. The implication of this is in the splitting of this branch point over here. So this is a metabolic pathway for purines and pyrimidines, and you have these distinct regulatory motifs. You can see the red motif, for example, appears only in this branch here, not in this branch here. So when that motif becomes active, it likely breaks this pathway and sends flux more in this direction at the metabolic level. This is all what I'm describing is at the transcriptional level. So just at the transcriptional level, by defining dynamic modularity, you can capture some of the evolutionary shaping of regulatory mechanisms to facilitate this adaptive processes depending on environmental context. So now this doesn't happen at a small scale, it's at a global scale. So hopefully this movie is visible here. So this is Halobacterium after irradiation with gamma rays. And you see a large numbers of genes are upregulated in yellow and a large number upregulated in blue. And they're connected to each other in different colored edges and the color of the edge indicates membership in corems. So you see different corems are operating in the downregulated gene set and the upregulated gene set after gamma radiation. But now you subject this to a different condition altogether, it's a late stage of growth. And what you observe is the regulatory connections operating in that condition are very different from what we're operating in gamma irradiation condition. And the whole network topology shifts, and you have corems that have broken apart and come together to form new structures. And was there a question there? Sorry, I heard someone. So do pause and stop me if you want to clarify something. So here, essentially, you're seeing the modular architecture of the same organism in two different contexts. And you see the modularity is dramatically different. And this is all biologically meaningful. What is the meaning of this in the context of metabolism? So here's an example from our earlier work where we show that there's a bunch of genes across these about a dozen biclusters that are regulated by this complex regulatory scheme. And you look at the membership of the genes across pathways, and just like I described in the previous example, the genes of these pathways are across multiple branch points. And you can see the same gene can be part of different biclusters. And this describes the dynamic modular changes that are required for plasticity in the level of vitamin B12 biosynthesis, in the context of pyruvate metabolism and peptide transport. Now, from a human perspective, this is very complicated to understand what this means at the level of phenotype. And to do that, and the scientist from my lab, Rupa, is there at the meeting. She joined my lab and she developed this really powerful approach of predicting the consequences of that complex regulation at the level of phenotype through integration of regulatory influences from transcription factors on enzyme coding genes that were associated with specific reactions in a constraint-based metabolic model. So we can take a model like this, and you can talk to her more about the technical details. And you can essentially use it to do flux balance analysis simulations of different knockouts. And you can predict which transcription factors or genes are important for adaptation to a given context and how that conditional importance changes as you transition from one context to the other. These are quantitative predictions that have been validated with genome-wide CRISPR screens and targeted experimentations. And I'll give you a vignette there and end with this vignette. And I picked the example of dynamic cell wall remodeling in TB. And I'll give you some quick background. So TB is the world's biggest infectious disease killer. It kills about 1.25 million people every year. And there's 10 million new infections. The current treatments are about 4 pills that are required to be taken in different combinations for 6 months. And even after that, 5% of the people don't get cured. And because of that, you have resistant strains that are emerging. And these strains are not responding to any treatment. And treatment of resistant TB can take up to 30 months with 10 different drugs and a period of up to 30 months. And the cure rate is just 56%. So it's a big problem. One of the major reasons we've struggled in developing drugs for TB is because of their lifestyle. And so here, what you're looking at is the house in which the pathogen lives. It's inside a lung, and this is called a granuloma. It's like a solid tumor. And it is composed of both host cells and pathogen cells. So there are host cells of different subtypes, different immune subtype cells. There's B cells and macrophages and T cells. And the pathogen lives in cells. Human cells have different subtypes, and also in epithelioid cells, and they can also be found in caseum in later stages of the disease. And each of these environments are associated with distinct challenges. So there's nutritional limitations, there's low pH, there's low oxygen. These are all efforts by the host to kill the pathogen, but the pathogen has learned how to live and thrive in this environment. Now we have to develop new drugs, we have to understand how it does it, how it is able to dynamically shift states from one microenvironment to the next to escape treatment and go on to get resistance. And so the conventional approach of using one or few drugs a long time ago, people figured out does not work because there's heterogeneity in the population, and you are unable to kill all the heterogeneous subpopulations with just one or few drugs. Then that's why you use 4 drugs or 10 drugs. And even then, we are not successful in treating some cases. So we need a rational way of understanding the mechanisms that generate this heterogeneity, and then in a model-driven way, identify drug combinations that can be uniquely tailored to a patient based on the lineage of MTB, the stage of the disease, and their other health backgrounds. So what we did is we compiled a compendium of about 2,000 odd transcriptomes, and these came from our own work and also from work done in 30 different laboratories. There's 53 different studies. And we took that data set and we generated the regulatory network model, similar to how I've described to you so far. And then we can mine this model for how the organism is able to remodel its cell wall and cell envelope. Now, this is considered one of the most, if not the most complex cell walls that evolution has ever constructed. And so what you have here are multiple layers of peptidoglycan, arabinogalactan, arabinomannan, and then you have long-chain lipids, and you have these special fatty acids called mycolic acids. And it's this waxy layer is what makes mycobacterium so difficult to treat. Drugs cannot make it into this through this cell wall and cell envelope. And what it also does is, and each of these layers is manufactured by a complex set of expanded family of enzymes. And they can mix and match different enzymes to change the composition of the cell wall, and they do that dynamically. And there's a cyclopropanation ring at the very end here that is very critical and controlled by 2 enzymes that are expressed in very specific contexts of growth. So how is this all managed by this pathogen? And where does this regulation occur to change cell wall composition to be tailored to the unique challenges of a given microenvironment? And this is where the ensemble modeling helped enormously. We were able to find regulatory mechanisms that act in very specific contexts. A MADR is a regulatory system that acts when there's fluctuations in oxygen tension. MTRA is a regulatory system that is triggered by changes in pH. And these are 2 of 82 different transcription factors that can mediate this kind of remodeling. But we know now mechanistically exactly what genes they're controlling and in what context. So we can design experiments to targeted inducible CRISPR knockdowns to show that, as the model predicted, they are really important for certain adaptations. And here I'm just showing you as an example, and I'll come back to this, is we're treating with 2 of the frontline drugs, isoniazid or rifampicin, at sub-inhibitory concentrations. And what you see is that concentration in the wild type, with and without induction of this non-targeting RNA, you see no effect on growth and killing. Okay, but if you induce the knockdown of this one is for MTRA, it completely clears the pathogen at the same dose. Right? So this is a model-driven discovery of a regulatory mechanism that operates in very specific host microenvironments that is critical for the pathogen for adaptation to the host. And we can now find how it's evolved this, and with a very precise approach design drugs. The MADR, since we discovered it about 10 years ago, has become the most important drug target in the field of TB drug discovery. And this is, again, a subset of the regulatory network that I was describing. So you have a set of transcription factors, and I'm showing you just about 6 or so here, that are regulated in a context-dependent manner by different host-relevant stresses or antibiotic treatment or inside macrophages. And then they go on to modulate the expression of all these genes across the different layers of cell wall metabolism. So arabinogalactan, mycolic acid biosynthesis, and so forth. And PDIM is a virulence lipid. And this is turning out to be a really important lipid in the cell wall of the pathogen that seems to govern its success or infection success. So using the model that Rupa has developed, she was able to then just take transcriptome profile information to then predict all the conditions in which these regulatory this regulatory network is essential for adaptation of MTB while it's growing on different carbon substrates, as it's transitioning in and out of hypoxia, as it's infecting the host within macrophages of different lineages, different pH levels, or in treatment with different frontline antibiotics. So we can then use this, and you can see how complex the space of combinations are amongst drugs, and it becomes even larger if you consider the different contexts in which you want to try their combinatorial effects. But this allows us to identify the right combinations in which we can identify new drug targets and predict their synergy with other drugs. And this is the last example I want to show you in terms of how modularity has helped in kind of uncovering some of this complex biology. So this is the eGreen2 network model for Mycobacterium tuberculosis we published in 2023. It's a hairball like all our networks are, but there's structure to this. There's interpretable structure. So seen here in these boxes are corems, the co-regulated modules. And these co-regulated modules are composed of genes, and the genes are associated with different functional functions, or they can be associated with different fitness and different environments. And the regulation of genes, and I'll give you one example. So down here is corem 71. Corem 71, the model predicts, is regulated by this regulator here, this triangle, and that's MTRA. And MTRA is predicted to regulate corem 71 binding to GRE11. So I pull that down here in this little gray inset box here. So in acidic pH, so this is the environmental influence in which this regulatory event occurs. In acidic pH, MTRA binds to GRE11, controls genes of corem 71, and the model predicts these genes are involved in cell wall and cell processes, so essentially cell wall metabolism. Okay. And then we can look to see in genome-wide fitness screens that indeed, in acidic pH, these genes show high level of importance. But we can use this to generate a series of hypotheses that are experimentally testable. So for example, we can then ask, is there evidence that the transcription factor actually binds to this gene regulatory element in the promoter as the model predicts? And these GREs were computationally derived using the approach I described earlier. And what you find here is that the gene regulatory element consensus motif that was discovered has got a lot more information content and is very significantly similar to the experimentally mapped cis-regulatory motif from ChIP-seq analysis, from two different ChIP-seq studies. And you can see in those studies, the information content of the motif is not as great. And the reason for that is they've grouped all the targets into one big group of genes or promoters on which they've done this motif discovery. Whereas what we've done is we've subsetted the genes that have evidence of being conditionally co-regulated and looked at the conservation of motif in their promoters. And that's the reason we get significantly more information content here. And then we can look at some of the functional consequences of regulation. And here we can see that the genes of these corems are indeed differentially regulated. When the organism goes from high pH to low pH, they get repressed. And this is part of a two-component system. So MTRA is a target of MTRB, which is a kinase, and it has two phosphorylation sites on MTRA. And when you remove those phosphorylation sites, you diminish the ability of MTB to adapt at pH 7 or at pH 5.6. So the phosphorylation system that relays the pH information is encoded at two different locations, and we can get down to this level of detail completely in a data-driven way. And the consequence eventually on phenotype is that, remember, we said it's important for cell wall metabolism and cell wall processes. So we did the knockdown and looked at the consequence on cell wall division, and you see the cells continue to elongate, but they don't divide. And you see multiple septae. In fact, when the MTR is knocked down, you see cells with up to 6 septae, which is typically never seen in wild type cells. And the consequence of that is it changes the permeability of the cell wall. And this is the reason we showed that drugs, regardless of mechanism of action, can now enter the cell and kill the pathogen very effectively. Okay? One vignette of how you go from large-scale multi-omics data sets to very precise mechanistic explanations of adaptation down to translation to drug discovery. So key takeaways, and then I'll stop. And I think we should have plenty of time for questions. So my presentation, my thesis was that based on the evidence we generated and other people have generated, and I heard this, I think Herbert mentioned it on the yesterday as well, that modularity is a dynamic object. It has to be probabilistically defined, and we have to realize that modularity changes depending on context, and this is a product of our evolution. And there's a lot of unsolved challenges here, right? We made good headway here. I think we have to recognize that this type of modularity is not just restricted to transcriptional and metabolic levels. There's modularity at the level of signaling at post-translational levels, and so on. And I've given the example of cutting across transcription and metabolism, but the same principles could be applied, to other levels of regulations as well. And by looking at the evidence-based change in regulatory mechanisms, you have a better chance of understanding phenotypic plasticity in a way that will allow you to then do rational perturbations. And I gave you one example of how we applied this to TB. And the effort, we're right now working with the Gates Foundation in particular to help characterize drugs of new novel mechanisms of action, and also predict combinations of drugs that are likely to be synergistic and the context in which the synergy is going to manifest. We're also helping them figure out ways to understand when and how the pathogen escapes treatment and what are the mechanisms that we can target to prevent that escape. We're also applying this to different types of cancers, where you can look at the modularity that underpins disease progression dynamics of patients and use that information to, one, predict whether they're going to respond to standard of care treatment, or if they don't, what other treatments are likely to be effective on those treatments. And this is being translated in the clinic as we speak. So there is a direct connection between understanding modularity at a mechanistic level to being able to translate the implications of that in a clinical or an environmental context. And a bunch of people have helped. I've kind of gone through the list to attribute their contributions to specific efforts, but I'll just highlight for the time being Rupa, who's in the audience right now. And she has a poster this evening on the PRIME model, which is integration of transcription and metabolism. So I encourage you to go and have a deeper conversation with her. So with that, I'll stop here. Thank you.
Yeah, so I'm wondering to apply this this approach you've outlined, how much transcriptomics data do you need? Like how many different conditions, and is there any kind of, I don't know, benefit to having like intermediate conditions, or is it better to just like grab the most extreme varied conditions you possibly can?
It's a great question. I think I think you touched on some key points that go into deciding what the data set should look like. When we built the first model, actually, we just had 400 to 500 different conditions. That was the E-Green model. The E-Green 2 model, we had about 2,000 conditions. And you're absolutely right. I think the composition of experiments in that compendium are important. So you need to have a diverse set of ecologically relevant insults that are represented. So in the case of TB, we had oxygen, pH, nitrosative stress, different host environments, carbon source. So there's a number of different conditions. And for each of those contexts, especially for drug treatment, for example, we had also different doses of the treatment. So you had, because you have very different effects at different doses. So you have essentially a dose response curve, and we sample different locations of the curve to see how the pathogen responds to those different doses. And then we take the shoulder of the curve, which is essentially where you see a phenotypic consequence of the treatment. And then we do a time course analysis on that. So we capture the time course transcriptome profiles, pre-treatment and then post-treatment over an exponential, a non-linear time course, essentially. So you go, because transcription is non-linear, most of the events happen early, but there's some events that continue towards mid and late state. So we go 5 minutes, 10, 20, 40 minutes, 80 minutes, 160 minutes. So that gives you a good space of diversity of perturbations and dynamics of response as well.
Questions?
Sorry, quite a naive question. Thank you for your very impressive results, especially as you translate it to real situations like TB. Just a question about the model itself. It wasn't clear to me whether this is predominantly an observational, like when you put to have all of these conditions to the left and then you see their consequences on the right side and you have 200 or a few thousand, whether this is predominantly something that you've observed or whether there was some other kind of process that you employed, some sort of regression or some other techniques to actually refine it. Because I'm not sure you could go through the entire space of conditions, right, to make, to get your model to predict really anything at all. So can you just expand on that a little bit?
Yeah, so certainly I think there's the observe part. The observe part is we're observing responses to carefully administered perturbations and how the system responds. And when we try to explain the dynamic changes over space and time, we are taking into account known causal mechanistic principles of biology. And there we're taking into account a prior knowledge of how transcription factors bind to promoters to either activate or repress genes and where they might bind. So we use that as a information prior to then guide algorithms to look for those signals, conserved signals, upstream to genes that now we have evidence of being co-expressed. Now, if they don't have evidence of co-regulation, then there could be two different mechanisms operating at the same time on these two different genes. And I've shown you a couple of examples of that. But once you identify these wirings, you can further reinforce it using ChIP-seq data. So independent, you can use ChIP-seq data for validation of the inferred influences, and you can upweight the ones that have evidence of physical binding. But you have to remember, physical binding maps are also incomplete, and they have a lot of false positives and false negatives. And there is a regression component here. Now, the regulatory influences you have inferred, one is mechanistically based on discovery of the regulatory elements. The second is through regression analysis using the interpolator, which essentially makes the assumption that transcript level changes of the transcription factor itself are predictive of transcription factor changes of downstream genes. Now, this may not be always true. Transcription factors may not be regulated themselves in a similar manner, but in many cases they are. So the regression approach does give you weights on the influence of the transcription factor individually and in combination with other transcription factors. Now, if you have ChIP-seq data, and if the transcription factor levels don't change by themselves, you can calculate something called transcription factor activity. And this was done by Mario Arrieta when he was a graduate student with Rich Bano. And Rich and I originally developed this whole framework. So he essentially took ChIP-seq type data sets and identified targets of transcription factors that don't themselves change, but based on the changes of the target genes, he reverse engineered the likely transcription factor activity changes. So there's many different ways that you can build quantitative influences that can be modeled to differential equations. And then when you're making predictions, the predictions are in new context where all you need is the measured levels of the transcription factors and environmental factors that can predict changes at that point in time. And we've shown you can also time evolve responses up to 24 hours if you have the right kind of data sets.
Thank you.
I have a question. How much did your modules discovered computationally match expectations from sort of traditional kind of pathways and traditional approaches?
There's a lot of overlap. And I think the point I was trying to make with some of the examples of purine metabolism and pyrimidine metabolism is you do recover known co-regulation among genes of a similar pathway. But there's more exception to that rule, which is observable in different contexts. So you see splitting of operons, you see splitting of pathways, you see reversal. I think there's an example in Rupa's poster where we show that there's a new drug that GlaxoSmithKline has discovered. They don't know how it works. They know it blocks cholesterol metabolism. And what we discovered was the reason it was being blocked is because there's an important pathway through which cholesterol gets utilized. That pathway, the gene expression has not changed at all, but two key genes downstream to that pathway have become overexpressed. And because of that, and some other changes in the cell, the pathway through cholesterol is metabolized, gets reversed. The reversal of the methyl citrate cycle cannot be predicted at the level of regulation. So that pathway unit is still the same pathway unit. Normally, it's co-regulated with upstream and downstream genes, but in these contexts, when you treat with a drug, it reverses its orientation and blocks cholesterol utilization. So when you think of modularity, I think it's not just which genes are together, it is what they're doing and who else is doing it with them. And there's also an aspect of directionality that transcriptional regulatory networks do not capture that is important for metabolism.
Thank you. Anything else? All right, let's thank Nitin again.